Histology of the small intestine. The same four layers that make up most of the gastrointestinal tract: mucosa, submucosa, muscularis, and serosa. Simple CA with many types of cells. Goblet cells which separate mucus. Many deep crevices lined with glandular epithelium form the intestinal glands or crypts of Lieberkühn and secrete intestinal juice. Panes and enteroendocrine cells. Panes cells secrete lysozyme, a bactericidal enzyme. Three types of enteroendocrine cells are found. These are SC cells, CCK cells, and K cells. They secrete the hormones secretin, cholecystokinin, and glucose-dependent insulinotropic peptide. The lamina propria contains areolar cavity and has an abundance of mucosa-associated lymphoid tissue or MALT, solitary lymphatic nodules, and aggregated lymphatic follicles. Pairs patches are numerous in the ileum. The submucosa contains duodenal or Brunner's glands secrete alkaline mucus. The muscularis consists of two layers. These are the outer thinner longitudinal and the inner thicker circular fibers. Lumen of small intestine. This is a diagram. Structural features of the small intestine facilitate the process of digestion and absorption, include circular folds, villi, and microvilli. Circular folds. Plicae circularis are folds of the mucosa, and the submucosa are about 10 mm long millimeter from proximal portion of the duodenum to mid portion of the ileum. Enhance absorption by increasing surface area. Villi or tufts of the hair are finger-like projections of the mucosa, 0.5 up to 1 millimeter long, increases the surface area to absorption and digestion. Each villus is covered by epithelium. Microvilli, projections of the apical membrane of the absorptive cells, is a 1 micrometer long cylindrical projection form a brush border extending into the lumen of the small intestine. About 200 million per square millimeter of small intestine greatly increases the surface area of the plasma membrane. Larger amounts of digested nutrients can diffuse into absorptive cells in a given period. This is the diagram. Large intestine is the terminal portion of the gastrointestinal tract. The overall functions are the completion of absorption, the production of certain vitamins, the formation and expulsions of feces. Divided into the cesium, colon, rectum and anal canal. The cesium or cecum is the dilated pouch that hangs inferiorly below the ileocecal valve. The appendix or 9 centimeter is attached to the inferior margin of the cecum or cesium. It has an abundance of lymphatic tissue which serves to resist infection. The colon. The colon consists of ascending or ascending, transverse, descending, and sigmoid portions. The ascending or ascending colon extends from the cesium or cecum along the right abdominal wall to the anterior surface of the liver, bends sharply to the left at the hepatic flexure or right colic flexure. Transverse colon crosses the upper abdominal cavity, bends at a right angle called the splenic flexure or left colic flexure. The descending or descending colon passes inferiorly along the left abdominal wall to the pelvic region. It bends medially from the pelvic brim to form an S-shaped curve called the sigmoid colon. The rectum, terminal or 20 centimeter portion of the gastrointestinal tract. It is last 2 to 3 centimeters. Is called the anal canal. The anus is the external opening of the anal canal. Two sphincter muscles guard the anal opening. These are the internal or smooth muscle and the external anal sphincter or skeletal muscle. The mucous membrane of the anal canal, arranged into longitudinal folds called anal columns, contain a network of arteries and veins. This is just the diagrams. Histology of the large intestine. The same wall layers with other gastrointestinal tract, mucosa, submucosa, muscularis and serosa. The mucosa consists of simple CE, lamina propria, muscularis mucosa. The epithelium contains mostly absorptive and goblet cells. The absorptive cells function primarily in water absorption. Both absorptive and goblet cells are located in long, straight, tubular intestinal glands or crypts of Lieberkühn. Solitary lymphatic nodules are found in the lamina propria of the mucosa. There are no circular folds or villi, however, microvilli are present on the absorptive cells. The muscularis consists of an external layer of longitudinal and an internal layer of circular smooth muscle. Portions of the longitudinal muscles are thickened, forming three bands called the tenia coli. Tonic contractions of the bands gather the color into a series of pouches called haustra. Omental or fatty appendages, small pouches of visceral peritoneum filled with fat are attached to tinea coli. These are the diagrams, layers of large intestine. Accessory digestive glands aid in the chemical breakdown of food. These are the liver, gallbladder, and the pancreas. Liver and the gallbladder, heaviest gland of the body, 1.4 kilogram, inferior to the diaphragm, right hypochondriac, part of the epigastric regions. The gallbladder is a pear-shaped sac, 7 to 10 centimeters in length, hangs from the anterior inferior margin of the liver. Anatomy of the liver and the gallbladder. Covered by visceral peritoneum and dense ICT layer deep to the peritoneum, divided into two principal lobes. These are a large right lobe and a smaller left lobe by the falciform ligament. The quadrate and caudate lobes belong to the left lobe. In the free border of the falciform ligament is the ligamentum teres or round leg. Remnant of the umbilical vein of the fetus extending from liver to umbilicus. The right and the left coronary ligaments are narrow extensions of the parietal peritoneum that suspend the liver from the diaphragm. The parts of the gallbladder, fundus, body, and the neck. Gross anatomy of the liver. This is the diagram of the liver. Histology of the liver. Histology of the liver. Histologically, it composed of several components. These are 1. Hepatocytes, 2. Bile canaliculi, 3. Hepatic sinusoids, hepatocytes, major functional cells of the liver, perform a wide array of metabolic, secretory, and endocrine functions. Are specialized epithelial cells with 5 to 1 to or 12 sides, 80% of the liver. And second, bile canaliculi, small ducts between hepatocytes. It collects bile produced by the hepatocytes. Bile canaliculi. Bile ducts. Bile ducts. Right and left hepatic ducts. Common hepatic duct, which joins the cystic duct of the gallbladder to form the common bile duct. 3. Hepatic sinusoids. Highly permeable blood capillaries between rows of hepatocytes. It receives blood from branches of hepatic artery and hepatic portal vein. Converge blood into central vein. Hepatic veins. Inferior vena cava. Kupffer cells. Phagocytes present in the hepatic sinusoids. Functions of the liver. Synthesis, storage and release of vitamins. Synthesis, storage and release of glycogen. Synthesis of blood proteins. Phagocytosis of all the red blood cell and certain bacteria. Removal of toxic substances and production of bile. Bile contains the following constituents. 1. Bile salts or bile acids, 11%. 2. Bile pigment, bilirubin, 1%. 3. Other organic constituents, cholesterol, lecithin, protein, etc., 3%. 4. Electrolytes. These are sodium, potassium, calcium, chlorine, and hydrocarbon or HCO3, 1%. 5. Water, H2O, 84%. Also takes the higher share of bile. Histology of gallbladder. The mucosa consists of simple columnar epithelium arranged in ridge. Lacks a submucosa. The middle muscular coat consists of smooth muscle fibers. Outer coat is the visceral peritoneum. Function: storing and concentrating the bile. Anatomy of the pancreas. Retroperitoneal gland, posterior to greater curvature of the stomach, about 12 to 15 centimeter long and 2.5 centimeter thick. 2.5 centimeter thick. Consists of a head, a body, and a tail. The pancreatic duct of Wirsang, joins the common bile duct and enters the duodenum as hepatopancreatic ampulla or ampulla of water. Thank you.